Good morning, Suzuki Science, grade 12s. I am happy to be presenting to you on our fourth week of learning outside the classroom. I'm hoping that these video lessons that I've been doing have been worthwhile. As opposed to just working your way through the PowerPoint, I've been trying to step you through with some of the same kind of conversation that I would be doing in class with you. Of course, we are cutting out lots of material because we are, believe it or not, abbreviating much of the learning so that we can keep it to a minimum. And then hopefully next year when you walk into your university, you'll be okay, because there'll definitely be some gaps, but we're trying to get the big ideas for you. Last week, in genetics so far, we focused on a number of things. We started off with replication, then we went into protein synthesis, and unfortunately we didn't get an opportunity to focus on biotechnology, which would have been a big part of the in-class experience. This week we're looking at genetic control, and next week we're looking at a little bit of biotech with your evaluation. So this week with genetic control, before we quite get there, there is a little bit of gap filling that we're going to do, and that is around genetic mutations. I'm going to switch back and forth between the PowerPoint. For the PowerPoint, mostly I'm just going to step through it. Like I did in class in the previous unit, I'm going to use another document in order to teach you the concept, because the concept is a little bit more abstract this week. But before we get there, let's get to the straight knowledge. Gene mutations. So in grade 11, we learned a bunch of things about mutations. We learned that mutations can occur, and those mutations that we talked about in grade 11, we actually talked about in grade 9 first, but let's back up year by year. In grade 11, we talked about chromosomal mutations. The idea that the chromosome could separate incorrectly in the form of non-disjunction, as you see on the page here. Right? A non-disjunction, we could see this happen right here. Right? Or, so that non-disjunction could happen during meiosis 1 or meiosis 2, producing a trisomy or a monosomy like Down syndrome. We also could have something happen to the chromosome itself. You have a normal chromosome from A through N, the genes, but sometimes you might get an inversion or a deletion or a duplication. And each of these are chromosomal mutations. They typically lead to, if the embryo or the young child is actually born, there's typically disastrous, catastrophic mutations that lead to catastrophic developmental processes, if anything even occurs. In grade 10, we looked at a different type of mutation. We looked at cancers. So in grade 10, we looked at the idea of cancers. We spent a lot of time on cancers. Here's the grade 10 slideshow. I just have it in the grid format so you can see some of the information that we would have covered in grade 10. However, we learned in grade 10 that a cancer can occur, and that's typically due to a proto-oncogene or tumor suppressor factor, something that controls the cell replication process, failing. There's a lot more to it, but at the minimum, that is some of the stuff that you'd learned in the grade 10. So that is also a genetic mutation that can occur. In grade 12, we're focusing on the molecular aspect of the gene. And we're looking at the nucleotides and the fact that a nucleotide might go awry. In the PowerPoint, you'll notice, if I were to step through, that I talk about different types of mutagens. These were talked about in both grade 10 and grade 11, and these are things that can cause a mutation to occur. Carcinogens is a general class, but there's also very specific types of mutagens that might cause a mutation to occur. Oncogenes and tumor suppressor factors, there's a link there that will help you to get more information if you want to review that. I will not ask you any specific questions on our abbreviated course about those. So I'm going to focus on the genetic mutations that are appropriate for grade 12. And that's going to be looking at the, oops, I went too far ahead. That's going to be looking at the nucleotides and how they're affected. So before I get there, let's take a look at where these mutations can occur. So looking at this diagram here, I'm just going to zoom in a bit because I can't see it that well, so I don't know how well you're going to be able to see it. At the DNA level, the DNA is first transcribed into an RNA, hnRNA first. In that transcription process, it is possible that a mutation may occur. This is known as a transcriptional mutation. Another mutation can occur after transcription has occurred. So when you're going from the hnRNA to the mature mRNA, you might get a mutation. And that is known as a post-transcriptional mutation. Then you have translation, where the RNA is translated into a protein. And in that step, you might get a mutation. That's called a translational mutation. It's translational control. And the final step is after the translation, we might get a mutation, some defect, something happens when the amino acid polypeptide is rearranged into its final sequence, post-translational. So these are all places where mutations can occur. We are going to be focusing on in this area, in the DNA itself. That's where our focus for grade 12 is. But recognize there's a whole world of mutations. There's so many mutations, as a matter of fact, that I don't necessarily want to teach. I'm giving you a worksheet. So for your week this week, for the grade 10s and 11s, we've always been giving them Google Sheets that they kind of complete, and as they complete it, they're learning their whatever they need to learn. For you guys, even though we're abbreviating it, pretty much everything that's still on the PowerPoint video lesson is you're responsible for knowing, unless we say otherwise. So in this document, I've given you this worksheet, and you'll see 18 different definitions and the answers. So you're matching the answers to these definitions. They're all related to mutations. In some cases, they're very specific mutations from grade 11 and grade 10. Some of them are more specific to grade 12. I have not taught it yet, but I will in just a moment. The answers will post up a bit later. And then as you continue in that document, you'll see some more things that will be relevant to the later part of the lesson. Okay. So in terms of mutations, let's go to our molecular mutations. So this is called a wild type messenger RNA. So this is a normal mRNA. It occurs in nature. Everything's fine. And if we look at the genetic code, remember our genetic code from last week, we know that each codon represents a very specific amino acid. So unfortunately, I didn't prepare myself by pulling up a genetic code, so it'll just take me just a moment here while I'm typing up on my other screen here. Genetic code. Oops, I misspelled that. I misspelled it again. Oh, I misspelled it again. All right, fourth time's a charm. And I'm going to grab a genetic code here, one that seems to work for us. That one looks good. So I'm going to copy that using my special copy application here. And I'm going to use this application just so I can keep it on the screen and bring it up back and forth for us. Okay, so here's my genetic code, so we can at least refer to that. So when I see GCU, I look for GCU on my genetic code, and I'll see that GC, right, U, so GCU is alanine. GGA is glycine, GCA is alanine, and so on, right? So we're just going to go through that. That's a genetic code. Everything's fine. But let's say we have a mutation. There are several different types of mutations that can occur. So as you go through my PowerPoint, you'll see that I've listed these things for you in a very sequential, orderly fashion that you can write down in your note. So I'm teaching the concept here. So looking at the first, let me reduce my size here. Zoom back to 200%. Looking at the first one here, we're going to call it a silent mutation. So look what we've done here. We're going to take this A that we have here of alanine, and we're going to have a mutation. So a single base tied mutation, nucleotide base that gets mutated. And so instead of an A, we now have a C. So my question to you, how does that change the overall reading? So GCA was alanine. So what is GCC? So GCC is also alanine. So what does that mean? It means that the same amino acid is coded for. When the same amino acid is coded for, that means even though we've changed the base and it's a mutation, in the grand scheme of the expression, there is no change. So the same amino acids put in, so there's no overall change to the overall protein, and everything's fine. So this is known as a silent mutation. Silent mutations can happen in a variety of places. Imagine a silent mutation occurring in a place on the intron, knowing that the intron gets junked out and tossed away. That's another place for a silent mutation to occur. Or let's say an aspartic acid is replaced by glutamic acid, and you just need an acid in there in order to react with something else along the chain. That could also be a silent mutation. So as long as the silent mutation has no effect on the protein expression, then we're fine. All right, so let's go back to our wild type, and this time, instead of having Let me get rid of that there so you can see. So instead of having a GGA here, that G gets mutated, and now we end up with a in that spot, right? So as AGA instead of GGA. So if we look back at our genetic code table, AGA is arginine, and arginine is not the same thing as glycine. They have different properties. So this is a problem. We've now changed the amino acid to a different one with the different properties. So that can cause a problem. This is known as a missense mutation because it still makes sense. The protein will be produced, however, it won't make the right sense. So we call it a missense mutation. Let's go to another one here, right? So in this one here, instead of having CAA, the mutation now, that C ends up being a U. Okay? And if you recall from your practice with the transcription translation from last week, that UAA is a stop. So the whole thing is going to stop right here. It's done. So now it no longer makes any sense. It makes no sense. It's a nonsense mutation. Each of these three mutations I've shown you here represent a point mutation. At a particular point in the sequence, we have a mutation. And in this case, we actually had a substitution. A C for an A, and an A for a, what was it, G, and here a U for a C. So these are all substitution. So we can actually have different names for the same mutation, right? So these silent mutation, missense, and nonsense are all point mutations, and they're all substitutions. So everything's fine. In our understanding, I hope. Now, in addition to having this swapped, we could also have a mutation where there's something inserted or taken out. And these mutations here are called frame shift mutation. So look at this one here. So in this mutation, if I take my wild type and bring it down, get rid of this, I have GCU, GGA, GCA. And right there where that A is, a C got inserted. And as you put a C in, it actually caused the entire reading frame to get shifted down. And so this is known as a frame shift mutation, and it is the result of either an insertion or a deletion. Okay, so as you look at the PowerPoint, you'll see all this stuff referenced. I'm just talking about it, talking it through. Hopefully it makes some sense. So we have different types of mutations. We have our missense, we have our nonsense, and we have our silent. These all represent point mutations. We have an insertion or a deletion, addition or deletion, and they cause a frame shift. Frame shifts are not going to be good. If you have a frame shift mutation, 99% of the time, we're done, right? Because everything is messed up from that point onwards, including the stop codon. It's not going to stop where it's supposed to. So these are some of the mutations that can occur. If a mutation does occur, then you need some way to fix the problem, right? So remember that you have a lot of DNA. And as the DNA is replicated, mistakes can occur. Even if you have a 1%, if you have 1% of 100, what is that? 1, okay. 1% of a million, 1% of 10 million, 1% of a billion, that's huge. The error rate on DNA replication is actually quite low. We're talking about 0.00 or 1% or something like that. But regardless, mistakes do happen. So we need something to fix that mistake. And you are going to click on this link, follow it through, and learn about DNA polymerase II. DNA polymerase II, with the help of ligase, is another exonuclease that allows the polymerization of DNA. But specifically, it takes out a mistake when it finds it. It uses one template to understand what's supposed to be there, and is the other template to fix the mistake. So click on these links. There's 2 of them, and they both together do an excellent job of explaining DNA polymerase II's role in scanning and fixing the DNA. All right, so that's all you. You'll also see the information about transcriptional control, and you'll see a link to this. I'm not going to show this during my presentation, but I encourage you when you open up my PowerPoint to click on that link and get some understanding. Why? Because that leads us to the next concept. All right, this concept is known as the operon. The operon is a part of the genetic control. The operon is used to allow the cell, whatever cell we're talking about, to clump all the units needed to produce a particular protein or to run a particular sequence of events together. Imagine you were going to run your car. If you want to run the car, you want to put your key in the ignition and you want the engine to start, the lights to come on, the heat to come on, you want them all to come on at the same time. You don't want to put in 3 different keys and 3 different spots in your car, one by your steering column, one underneath your seat, one in the back seat. That would not make a lot of sense. You put one key in one spot and everything comes on together. That is how the operon model works. So here is a little graphic I have, and I'm going to explain to you the different parts of the operon. This is a model that's found in a prokaryotic cell. We are going to avoid talking about eukaryotes because they are far more complex, but the same rules generally apply. So here are the parts of an operon. The operon is made up of the genes that are to be expressed. So if you want to produce insulin, if you want to produce a signal, if you want to produce whatever it happens to be that's a protein, the DNA is transcribed and translated into a protein. That DNA that codes for something, these are called the structural genes. So structural genes are what you have been learning about. These are the story, right? This is the main part of the story. In this case, on this particular operon, I have 4 structural genes put together. All 4 of these structural genes might come together at the end to produce a particular protein or a set of proteins that will allow a process to occur. So if I want to produce muscle, and let's say muscle requires actin and myosin and some other things, I want to put the gene that codes for actin in the same place I put the gene for myosin so I can produce them both at the same time, if that is what I want to produce at the end. It wouldn't make sense to put 2 different parts of the gene that are 2 different genes that are responsible for the same thing in 2 different spots with 2 different keys, just like the car model. So for these genes, we need an on-off switch. We need something to say turn on or turn it off. And that's the operator. The operator is the on-off switch. I remember it because it starts with an O. The enzyme that's responsible for transcribing the genes into an RNA is known as RNA polymerase. The RNA polymerase has to bond somewhere, and we saw this when we started talking about the initiation event of transcription. We know it bonds to the DNA somewhere. That place where the RNA polymerase bonds on the DNA is known as the promoter. So the promoter bonds the RNA polymerase. It's like a big magnet. Think of this magnet here, it attracts the RNA polymerase as a magnet, and it comes together. So what I've just described here with the promoter, the operator, and the structural genes represents the bulk of the operon model. I mentioned this on-off switch. So you need something on that attaches to the on-off switch that will turn it off. And that thing that attaches to the operator to turn it off is known as the repressor. The repressor is typically a protein that bonds the DNA here, and it will physically impede the promoter, the RNA polymerase, from moving down. Okay, so this repressor, which is a protein, it also needs to have a gene that codes for it. It's a protein. All proteins have some gene that codes for it. So the gene that codes for the repressor is called the regulatory gene. It doesn't just have to be a repressor. It could be an inducer. We'll talk about that later. But this regulatory gene, which is somewhere typically upstream, remember this is the 5' end, it's the 3' end, so somewhere upstream, this could be several hundred base pairs, it could be several thousand base pairs, way up here, there'll be a gene that codes for the repressor. And then that repressor might bond to the operator, turning it off. Those are the parts of the operon. That is your operon model. This is found in the prokaryotic cell. You should be familiar with the parts. So next, I'm going to explain how the operon model works. If you go through the PowerPoint, you'll see, and I'll just go through it here, I give you step-by-step instructions about 2 very specific types of operons. One is an inducible operon, one is a repressible operon. Okay, so you can work your way through that to get all the details, and I'm just clicking through so you can see what's on here, along with some scientists that contributed to the knowledge. I will not ask you any questions about Jacob and Monod, but I will ask you lots of questions about the operon model. I'm going to stop this particular point at around 20 minutes. I'm at 1940, so that way I can just keep them in small little chunks. So I'm going to end this now, and when I come back, I'm going to continue with the operon model.